like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place.
Welcome to the National University of Singapore Yong Loo Lin School of Medicine Healthy Longevity webinar. Thank you for joining. As always, you will not be disappointed. We will learn from Dr. David Ferenbach how the kidney this time is aging and what role cellular senescence plays. Please, you might have lots of questions. Use the Q&A function to send comments, suggestions, remarks, and especially your questions, which we will answer. They are really highly welcome. As always, we are starting with a very short presentation for 5 minutes, this time by Si-Meng Lim, and he is research assistant at the Center for Health and Longevity, and he will present about his favorite topic, and that's the brain, summarizing an article titled Distressing Dreams, Cognitive Decline, and Risk of Dementia, a prospective study of 3 population-based cohorts. Si-Meng.
Thanks for your kind introductions. As mentioned, I'm Si-Meng Lim. Today, I will present an article titled Distressing Dreams, Cognitive Decline, and Risk of Dementia, a prospective study of 3 populations-based cohorts by epidemiology article published in eClinical Medicine, which is part of the Lancet Discovery Science. The background of this study was distressing dreams demonstrated association with faster cognitive decline and increased dementia risk in people with Parkinson's disease in previous study. But distressing dreams might be associated with cognitive decline and dementia in people without Parkinson's disease is unknown. Therefore, this study aims to investigate the association between distressing dream frequency and the risk of cognitive decline and incident dementia in community-dwelling men and women without cognitive impairment or Parkinson's disease. This is the longitudinal study, involved 3 different groups of people. Self-reported distressing dreams was assessed, and the participants just answer how their sleep quality. So author had came up with an equation to assess the cognitive declinations, and the whole study was comparing baseline results and the follow-up results. So table 1, baseline characteristic of middle-aged cohorts by distressing dream frequency, N is 605, and the table 2, the baseline characteristic of older adult cohorts by distressing dreams frequency. So distressing dreams were more frequent among women in middle-aged cohort, but were more frequent among men in older cohort, as shown in table 2 and table 1. The more often the subject experienced distressing dream, the higher risk of having cognitive decline among middle-aged adults. Also, it shows a 4-fold risk of experiencing cognitive decline for subject having more than 1 distressing dream per week. However, if they dig deeper into gender, this association only show among middle-aged men. Now, within the older O, a higher risk of dementia was shown. Also, it shows a 2.2-fold of difference in dementia risk among older adults. The author had many discoveries, but I will highlight a couple of them. Prevalence of frequent distressing dream once a week or more was higher in older cohorts in present study compared with middle-aged cohort. The result of the current study suggests that the emergence of distressing dream during middle and older adulthood may represent early sign of neurodegenerative dementia in some individuals. A limitation of this study is that it was conducted predominantly among Western cohort, so the results may not apply to Asian cohort such as Singaporean. Hopefully, we are able to assess it one day in our clinic. In summary, this study provides evidence that a higher frequency of distressing dream in community-dwelling adults without cognitive impairment or Parkinson's disease is positively associated with faster cognitive decline during midlife and increased risk of developing dementia during later life. As such, this study suggests that screening for distressing dream in the general population may help to identify individuals in the preclinical phase of dementia in whom early interventions to prevent cognitive impairment could be targeted. With that, thank you.
Thank you so much, Simeng. I think we should screen not only how good the quality and the quantity of sleep is, but now also talk about, what was it? Distressing dreams. I would call them nightmares. Thank you so much. We are moving from the brain and the brain sleeping now to the kidney, and we have two kidneys actually. Dr. David Ferenbach is a clinician scientist and specialized in nephrology and working also clinically in the nephrology department in Scotland at the University of Edinburgh. And his research is focused on the understanding of the mechanisms linking kidney injuries to fibrosis and premature aging, of course, and kidney decline in their function. And of course, the role of cellular senescence, which is very important. I just heard him during a conference last week at the International Cell Senescence Association annual meeting, a wonderful presentation he gave. So his aim is really to develop new treatments for human fibrotic disease, and especially in the kidney. How wonderful would that be? So David, thank you so much for joining today from Scotland. I think it's cold over there. The floor is yours.
Excellent. Well, thank you very, very much for that very kind introduction. It's an honor to be asked to talk today. And Singapore is a wonderful place. I've got very fond memories of visiting myself in the past. Now, what I'm going to be talking about today is, as you've just heard, is aging, senescence, and how both these processes may affect our kidneys. And in doing so, I want to try and tackle maybe three questions here to a greater or less degree. Now, obviously, I'm biased as a kidney doctor here, so I'm going to talk a little bit about what chronic kidney disease actually is, and then going to go on to talk a bit more about what cellular senescence is as well, and discuss why it might be that senescence impacts, in lots of organs, but in particular in aging and chronic kidney disease here. And just, if I have a couple of minutes at the end to talk a little bit about some of the many questions that we don't know the answer to just yet about senescence, but ones we're thinking about a bit in my lab at the moment. So I suppose the first question would be, is why am I, as a kidney doctor, talking here at a meeting interested in healthy longevity? So chronic kidney disease, the first thing about it is chronic kidney disease is a common condition. Over 700 million people worldwide have it. The large majority of them don't know that they do. It's common, and I'm afraid to say it's incurable as well. And preparing this talk, it just sort of struck me again how much we need better treatments for chronic kidney disease. Because although lots of different things can cause it, immune-mediated processes, hypertension-mediated processes, genetic processes, something they all have in common is that they cause progressive kidney scarring, which can end up with your kidneys not working at all, at which point you either die or you end up in some form of dialysis treatment or transplantation. And while we have blood pressure medicines, ACE inhibitors, in the last few years, we at last have SGLT2 inhibitors, the combination of all of these things doesn't stop this or reverse it. The best we can do is to attempt to slow it down for as long as possible. And another really important thing, I think, is that even in these hundreds of millions of people, most of whom will never need dialysis, will never see a kidney doctor, the trouble is, even if they feel fine, they have got a major elevation in their risk of morbidity and mortality, most of all through premature cardiovascular disease, such that various people have proposed that having chronic kidney disease is almost like a syndrome of accelerated aging in terms of how other pathologies seem to happen more quickly and across the body beyond the kidney. So when it comes to my interest in aging, and I've sort of deliberately pinched a picture of a really sort of healthy looking young patient here being attached to a dialysis machine. And even in this setting, somebody much younger than me, perhaps with a kidney limited disease like Goodpasture syndrome, which is now inert on dialysis. The trouble is you can dialyze and you can stop them dying of uremia. If you look at this graph here of the US population back in 2005, someone based in their 20s or 30s, which have actually on average, in the absence of kidney disease, about another 45-year life expectancy in America. If that same patient was on dialysis, their life expectancy is less than 10 years. In fact, you'd be worse getting a number of common malignancies and ending up on hemodialysis. It hugely reduces your quantity and quality of life. And in fact, even if you give this patient the absolute, what we would call the gold standard treatment, a well-functioning kidney transplant, that would very significantly increase his or her life expectancy and the quality of life as well, but it still gets nowhere near the general population again. So there's something about having a damaged fibrotic kidney which seems to impact permanently on your life expectancy and impact on other organs as well. And a question which has occurred to me and occurred to many other doctors is, could it be that cellular senescence is playing any role here in this progression of chronic kidney disease, progression of fibrosis, and even these systemic consequences? But to think about that, we also need to have a think about actually what is senescence anyway? Who can get senescence? And I've got a very broad answer to this because it's really anything, any multicellular organism. Even if you're a nematode worm and you only have 1,000 cells, some of them will be senescent. It happens to fish, to amphibians, happens to mice, happens to healthy young adults. In fact, actually, by the time all of us are born, we will have undergone waves of developmental senescence that has occurred and been cleared very quickly in a nice programmed way. But an area which I think is attracting lots of interest from researchers, from clinicians, and indeed from pharma companies, is the recognition that as we get older, we accumulate senescent cells across all the organs of our body. They accumulate even more in the context of illnesses. And there's now really good evidence that senescent cells are not just innocent bystanders, they seem to be contributing here as well. So what is a senescent cell? What makes a senescent cell different? You can sum up, I think, my knowledge of this as an undergraduate medical student was this: senescent cells don't divide anymore. They've undergone permanent growth arrest. But equally, lots of cells are in that setting and are not senescent. The other thing about senescent cells is they've got an altered transcriptome. These are cells that are behaving differently. It means they express lots of different genes, and they classically acquire this thing we call the senescence-associated secretory phenotype of releasing lots of potentially bioactive molecules into the sort of local and even systemic milieu there. So if that is what a senescent cell is, and given the huge number of organisms all have them, you feel we must have developed them and retained them for a reason. And there are good things that senescent cells do. A first one is this growth arrest, and this can occur in response to cell stress and to DNA damage. And actually, I'm very pleased that I have functioning senescence such that if a cell damages an oncogene or an oncosuppressor gene, a classical mechanism is to become senescent rather than becoming a tumor. And in fact, the molecules that control senescence are some of the most commonly mutated in malignant tumors. So senescence is a good thing here, I think. And indeed, when we talk about acute senescence being triggered in response to an injury, and the more research we do in this, we see that these senescent cells, when they switch on these signals, these can recruit leukocytes to phagocytose them and clear them. Sounds like probably a good thing. There have been some very nice studies looking at the role of senescence in injury, showing that actually having senescent cells will actually signal to switch fibroblasts on at the right time, and indeed to switch them off at the right time as well, to try and come up with an optimal scar. And there have been some studies looking at senescent cell deficient mice. It shows actually they have deficient wound healing as well. So again, more reasons why actually we should be happy to have senescent cells. But it's not all good. And with these mechanisms come potential bad signs as well. So when we talk about this growth arrest, there's this concern that as we age and accumulate senescent cells, if actually we are switching off our sort of pool of stem cells, or even influencing their behavior via this release of factors from senescent cells, this may be a bad thing. It may limit our aging organs' ability to recover after injuries. There's then, when we look at senescent cells that hang around and don't get cleared, what we call these long-lived chronic senescent cells, we accumulate these as we get older. We accumulate them with injury as well. What I should say is just accumulating something doesn't necessarily mean it's to blame. It doesn't necessarily mean it's bad. It could even be a good thing. But we certainly see more senescent cells with illness, with aging as well. And with their signaling, this seems to also result in a recruitment of inflammatory phagocytes as well. And certainly in the kidney, at least, that's not a good sign. That actually predicts bad outcomes there. And in lots of human diseases, when we quantify senescent cells, the more of them you have, the worse the disease seems to go. But again, you just have to bear in mind, senescent cells could at this stage certainly just be innocent bystanders, the marker of some other bad process going on there. So coming back to this question of whether cellular senescence might have anything to do with chronic kidney disease and systemic consequences, I suppose our first question is, are there even more senescent cells in chronic kidney disease kidneys? And actually, this has been answered years and years ago by other researchers. We have an excellent group in Canada. The group of Annette Malcolm, Roland Schmidt over in Hanover have looked at this a lot and have shown that, yes, you do get more senescent cells with renal aging and with kidney disease. This example here is one of their studies where they looked at a healthy young 19-year-old kidney. Up at figure A on the left here shows no evidence of brown markers of senescence at all when it was transplanted. When the same kidney was followed up and rebiopsied a year later because this kidney was deteriorating in its function, what they found was lots and lots of senescent cells present. So with worse function, you do often see more senescent cells. But again, the caveat is they could, again, just be, if you like, innocent bystanders from other unrelated injury as well. But it still is intriguing. And something actually as kidney doctors where our understanding is changing is what happens after the kidney is acutely injured. Because I think the textbooks will tell you, and it's true, the kidney is capable of repair after a sudden limited insult. You have a patient who comes into hospital, becomes very unwell for another reason. They get an ischemic injury to the kidney, in particular to the epithelial cells in the kidney. You can see cell stress, cell death, and these red cells in this diagram show what we believe to be senescent cells. But the thought is, if that kidney can be supported with dialysis and the patient kept alive, you will get adaptive repair. These acutely senescent cells will recruit leukocytes. Senescent cells will be cleared, proliferation will occur, and at the end of the day, you end up with a healthy, normal kidney again. And that is a theory, though bear in mind, we almost never biopsy people in this situation because kidney biopsies in this situation wouldn't help our management and actually could cause some harm. And what we proposed in this JASON article from 3 years ago here was that something may happen in a lot of other settings, in particularly older patients, in particular people with pre-existing kidney disease, with pre-existing senescent cells. We recognize that often after a kidney injury, they never get back to quite where they were before. And the thought is perhaps actually we're seeing a maladaptive repair here, and there is a possibility senescent cells might contribute to this. Could it be that having senescent cells sending out this SASP signals actually drive this chronic inflammatory infiltrate? Could they have paradoxically effects causing more fibrosis and leading to a kidney that works again but never works as well and actually progresses on that route of fibrosis that we most want to avoid? And as I said, this was a speculative article where we proposed this. We were very careful to cage it with potential there because we had no evidence that this was the case, or only circumstantial evidence. What did provoke us to do was to try and chase it a bit further. And this was work led by Katie Milonis, who was a research fellow in my lab at the time, looking for more evidence of senescence in the kidney. And we turned to publicly available data sets, looking for these major markers of senescence, the genes CDKN1A and 2A, code for the proteins P21 and P16 that are key mediators of permanent cell cycle arrest. And when we looked in these published data sets, comparing patients with chronic kidney disease to healthy controls being assessed for kidney donation, they've got significantly more of both of these markers, actually in particular, the gene coding for P21. And when we related these to the age of the patient and to their kidney function, it was actually P21 was the one that linked most closely. Both markers rise significantly with an aging kidney. P21, the more you have of it, the less well your kidney works as well. Again, this isn't proof, but it would go along with these two things being in some way related. And in ongoing work, David Baird has tried to take this a bit further within kidneys. We've got permission to access this sensor biobank we hold here in Edinburgh of kidney biopsies with linked clinical follow-up as well. And what David has spent a lot of time doing is staining these kidneys using our whole sort of slide scanner here over multiple images. And because he's a much better coder than I am, he has written some code that can actually identify where the biopsy is, allows to zoom up and up in it. And these lines here in yellow show what it's now identifying as tubular cells based on the expression of these key epithelial markers. And now, while David goes away and has a cup of coffee, the computer can analyze, look at this tissue, identify epithelial cells. What I hope I can convince you here is it can identify the nuclei as well. And in some cases, it's spotting red nuclei expressing this P21 marker of cell cycle arrest. In some, in marked white here, it shows that these cells are coincidentally proliferating and therefore have to be excluded. But what it can do is actually identify and classify cells that have the markers on are not proliferating, so fulfill one criteria of cellular senescence. And then, after analyzing over 50 human kidneys and counting many, many thousands of cells, it will read out the proportion of the epithelial cells doing this. And if you see between the left and the right, if you were just looking at this as a kidney pathologist, you might not see very much different between these at all without this stain. But this kidney on the left here, more than 1 in 5 of its epithelial cells senescent. The one on the right, less than 1% of them are. And when we go back to see how this relates here to the markers before, actually looking at epithelial cell protein levels of P21 correlate better than any of the other markers at transcriptomic level. They go up as our patients age, and the higher P21 you have goes along very significantly with reduced kidney function as well. Now, as for the graph, I must admit, I'd be most interested in. I'm afraid we remain short of patients for this, and we're in the process of expanding this cohort here. What I can say is on this graph, there is no significant difference yet. Although if you're a patient with low cellular senescence in your biopsy, actually you're more than twice as likely to still have a functioning kidney at the end of the study compared to those with higher levels of senescence. What we need is more numbers and more endpoints to try and tease out whether this is really an additional sort of prospective risk factor which to sort of tell our patients what the future may hold. What was happening in parallel with this, though, was that Katie Mylonis went back to mouse models to try and really tease things out a bit more. And what she used here was a model of ischemia reperfusion injury in a mouse. So we give a single mouse kidney a really significant injury, such that in the aftermath here, you see, I hope I can show you with my pointer here, a big permanent increase in cellular senescence, actually measured here by P21 staining. You see a big increase in fibrosis here as well, which is this nasty pink stain down at the thing at the bottom here. And what she compared it to was 2 other categories, 18-month-old mice, so really properly aged mice, and young mice that had undergone irradiation to generate senescent cells, despite the mouse still being young. In each of those cases, what we found was there were more senescent cells before injury and significantly more senescent cells after injury compared even to an injured young mouse. And along here again, this went along with more fibrosis for the same level of injury. So a same injury model gives you a pink or even worse looking kidney. But is this proof that senescent cells are doing this? No, it still isn't. It's an association. But what Katie did next was that she did go further and she used a drug called ABT263. And this is an agent which has attracted a lot of attention because it inhibits the BCL survival family of factors. And there have been some great publications showing that a lot of senescent cells rely on this pathway as a route to avoid undergoing apoptosis. So when we block this survival factor enriched in senescent cells, if you look at these graphs at the top here, she brought the levels of senescence right down in the baseline kidneys. You can see it there by the white staining up in the top left in the nuclei. And then in these baseline kidneys, we did nothing for a couple of weeks, allowed all the drug to clear the system. But then under post-therapy conditions or post-vehicle conditions, we then did the same injury. To try and answer the question is, do these old and injured kidneys still have this worse outcome? And the answer was no, they don't. If we have pre-depleted the senescent cells, an old kidney actually start to behave more like a young one again. Here you can see the pink scar you get after an injury is significantly less here in this image. And I think most importantly, your kidney function is better maintained as well. These old kidneys and these irradiated kidneys, again, they behave like young ones when you've cleared their senescent cells. They're better able to repair. And so at the end of all this, I think what we can say is that yes, senescent renal epithelia are increased with aging. They are increased with chronic kidney disease. And these cells are actually inhibiting this complete adaptive repair that we see after injury. And importantly, there's a potential to treat this as well. We can give drugs to kill them and actually seem to be able to, for the first time, perhaps make an old kidney behave a bit more like a young one as well. Now, another arm of work, which again was overlapping with Katie's work, was this work done by Owen O'Sullivan, who was a clinician doing his PhD in the lab. And again, we were asking this question again, was how well do kidneys recover and reverse fibrosis after an acute insult which has gone away? Because if you did, most of your textbooks, unless they're very, very recent, will probably say the kidney has a wonderful and complete ability to repair after injury. But with more studies now, I think that is wrong, in fact. When we follow patients up who have apparently been discharged from hospital with completely recovered kidney function, their chance of ending up in a kidney clinic afterwards with chronic kidney disease goes up by nearly tenfold. Their chance of ending up on dialysis is about 30-fold increase compared to healthy controls who've never had such an insult. So injuring a kidney does seem to have a long-term effect on patients, which is a very, very bad thing. And in fact, what we wanted to do is try and see what would happen if we injured and then allowed a kidney to regenerate in a mouse as well. And one of the supposedly best models of this is reversible UO, where a kidney is blocked, giving it this, for a period of time, a really severe obstructive injury where you recruit inflammatory cells, senescent cells, and fibrosis. However, if you then reverse the obstruction, then the insult is completely away, the leukocytes disappear, and the kidney starts to get better again. What we would say is when Owen and I followed this up long after injury and actually took kidney and analyzed it by RNA sequencing and looked at the pattern of dots here on the left. The first 4 bars there in gray are kidneys that have never been injured. The ones in orange are ones that are 5 weeks past removal of insult. And again, it seems to be pretty clear what you still have is many more markers of ongoing senescence late after removal of the insult. We have got more markers of SASP release still in many parameters, and collagen increase and fibrosis markers remain up as well. What's particularly intriguing is, though, if you injure these kidneys, allow them to repair, but dose them with ABT in the early aftermath and then follow them up weeks further down the line, they are much better, actually. What we see is reduction in senescence, in SASP release and fibrosis, and along with that, many, many pathways linked to this are all significantly up or down regulated too. And in fact, when Owen went on to do this and followed an RUO out that had had this treatment, what we see is a kidney which is much closer to normal again. There's still a bit of collagen there. There's still probably more fibroblasts in the baseline kidney. It's certainly improved with reduction in the number of senescent cells present there. So I would say it's further evidence that senescent cells are driving maladaptive repair and progressive fibrosis. Now, a really key question here is, how are they doing that? And to do that, we took advantage of new technologies coming online here. And we started doing this experiment again. And this time what we started doing was single cell RNA sequencing. It allowed us to pull out every cell in these kidneys and analyze them individually here. This is an example of every epithelial cell in a sham kidney. The orange and red dots are senescent cells, and there's very, very few of them. When you look at an acutely injured kidney here, we've got lots of acute senescence there, spanning particular subsets of renal epithelial cells. But again, if you look in that injured kidney and follow it out weeks down the line, the senescence reduces, but it never completely goes away. And taking the many, many genes we're able to analyze with this, we then compared this to other published data sets. This is a publication here in a kidney here by Ben Humphrey's group over in the USA, looking at a rejecting human kidney. And group B here was a published publication actually in Nature from some of our collaborators in Edinburgh, looking at the senescent cell and the total transcriptome cirrhotic livers. When we compared all these together, what we hoped is it might uncover factors that are conserved across organs and across species as well. So hopefully some of the core factors actually driving adverse outcomes in senescence. And indeed, by the time we'd done all that, we were down to 11 factors that seemed to go up significantly in senescent cells across all these 3 settings. To chase things further, we then took our epithelial cells in vitro and we injured them and looked in senescent ones to see if these factors indeed went up and down. This narrowed it down further. We still had several factors, which I hope you can see in the orange bars here are significantly up early and late after injury. And of all of these, when we then went to another online data set, to look at their behavior in human chronic kidney disease, it was PDIA3 which was most significantly upregulated with amazing p-value in human chronic kidney disease compared to healthy controls I looked at. So we then honed in on this and spent a couple of years chasing this in vitro and in vivo. And actually, at the end of all this, what I can say is it all comes back to TGF-beta, this master regulator of kidney fibrosis and indeed fibrosis in lots of organs. And the way TGF-beta works is it links to the TGF-beta receptor. And in doing this, it phosphorylates SMAD2 and 3, which go to the nucleus and drive fibrotic factors. But another thing that is absolutely essential for this is this factor called SARA, which actually mediates that phosphorylation step. If you don't have SARA, you don't generate phosphorylated SMAD. And what we discovered eventually after a lot of looking is that PDIA3 being upregulated and excreted by senescent cells is one of the things that drives SARA production. So in the context of more PDI3, you will have more SARA, and for every molecule of TGF-beta, you will actually phosphorylate more SMAD here as well to then translate to the nucleus and drive fibrosis. And with this, actually, if you do the opposite, if you can block this interaction between PDIA3 and SARA, what you can see here for TGF-beta over on the right here is that phosphorylated SMAD virtually goes away. And when we step back into our mouse model and gave it one of the small molecule inhibitors available to block PDI3. When we do this, we switch off pSMAD2 production in the post-injured kidney, and with that, we downregulate total mesenchyme, activated fibroblasts, and collagen production very significantly as well. So I would sum this up saying that our single cell analysis so far of renal epithelias has uncovered signaling pathways that we would never have thought to look at before. We are very limited in our ability to look at all of them. The one we did pursue actually looks promising. It may be a new target which we can perhaps target specifically to look at the generation and the contribution of senescence to fibrosis with aging and after injury. If I have time just to talk about a couple of future questions here. One thing I remain interested in is whether transgenic mice are capable of telling us more about cellular senescence. With this work, which has been led by Cyril and Mary, one of our scientific and medical PhD students here, we've generated a novel transgenic mouse here. And in this, we have built M-neon green, a fluorescent green protein behind the native CDKN1A gene, along with an inducible prey. And what all this means is that in these mice, every senescent cell expresses M-neon green and lights up green under a microscope. What we've also done is we have crossed this with the AI14 reporter strain, which then gives us the ability, by giving tamoxifen to this mouse, we will then switch on that inducible cray in every cell, leading to senescent cells being both green and red as well. So you can actually date stamp these cells in vivo using tamoxifen. It also means that after tamoxifen, you can still generate more natural senescent cells, but they will still just be green again. So you can differentiate older senescent cells from younger senescent cells. And if my computer lets me, I can show you, I hope, this next plot here. These, you have to take my word from it, are beautiful pictures of renal tubules. You can see these orange ones are expressing both red and green fluorescence. There's also bright green ones as well. So they're only expressing the M-neon green fluorescence. And the thing that actually intrigues me most about this is an injured kidney weeks after IRI, weeks after tamoxifen injection, and it's a kidney slice. This is the whole kidney. So what we're doing here is looking at this whole kidney using intravital microscopy. So my hope is this is a tool that will actually allow us to look at senescent cells in live mice and chart how they behave, how they're cleared, how they persist. It is all driven under the P21 promoter here, and the scope now to pull these cells out and look at them, to correlate their appearance and their behavior. I think there's a lot of scope to look at their kinetics as well. And lastly, with this inducible cray, there is a scope then to start chopping out genes in natural senescent cells under the control of tamoxifen to start working out what gene is doing what. Another question which we are trying to come up with an answer to is whether senescent is just intrinsically pro-fibrotic, even in the absence of being old, being acutely inflamed or deprived of oxygen, for instance. To do this, we have just crossed lots of other people's minds together to try and give us a tool to answer this. So what we've used is the inducible cray system driven by PAX8, a promoter which is really tightly restricted to renal epithelial cells. we've crossed this with this MDM2 flox mouse. Now, MDM2 is one of the natural regulators of the P21, P53 senescence induction process. So if you do inactivate MDM2, you get this P21, P53 driven growth arrest and senescence. And finally, we've added in this AI14 reporter again to allow us to actually chart the cells where recombination is taking place. And in this mouse, in the absence of tamoxifen, it is normal, it has no fluorescence, it has no phenotype. If you give this mouse tamoxifen, though, what will happen is just in these PAX8 renal epithelial cells, you'll switch on Cray. The Cray can go to the nucleus and will chop out these LOXP flank sites, meaning you lose active MDM2, you switch on your TD tomato. What this means is that you will then upregulate P21 and P53 in a subset of cells, and they will enter permanent growth arrest and senescence. I can show you in this graph here. Here is a kidney, actually two kidneys. One is a wild type, one is a transgenic. Both have had the same dose of tamoxifen. You can see these bright red cells appearing stochastically across the tubules here, but none in the other spaces there, showing recombinations occurring. And when we follow them out after injury, even at this early day 7 time point, what we see is a mild but very significant increase in fibrosis here. Now, you'll see no tubular injury here. There's no hypoxia or other insults, but things are different. And again, when we use single cell sequencing to try and pursue this further, we've collected over 22,000 epithelial cells here, spanning the entire epithelial compartment of the kidney. This big blue group here are your proximal tubular epithelial cells, over 14,000 of them. But that's not all. When we look at ordinary mice and our transgenic mice, in our transgenic mice, these are the only proximal tubular cells. These other populations here all are expressing proximal tubular markers, but you can see there's a space there. The computer, in an unbiased way, is identifying these cells as transcriptionally different. When we look at these cells, in all 3 of these clusters, they're expressing more CDKN1A compared to the rest of your proximal tubules. And as you see here, this is a classical marker, a prototypic marker of proximal tubular differentiation. All these cells express it. And when we look at other markers of other renal epithelial differentiation, they're negative of all of these. So these are proximal tubular cells, they're different. And within these subsets, they're different as well. So you've got very, very different gene expression between some of these subsets. The other thing I found fascinating was some of these subsets have deleted MDM2, what we have done, what we've induced in them, other ones don't. So already what we have here is a model where we can chart primary senescence, but also paracrine or secondary senescence as well. And what I hope is that these transgenic approaches in the future will allow us to find out more about exactly how senescent cells behave, how they signal, how they interact with each other as well. But as I say, there's many more questions there than answers, I'm afraid. And now it just remains for me to acknowledge all the work from my lab members in the collaborating labs that have made any of this possible, and the funders here on the right, Kidney Research UK, the Wellcome Trust, the Medical Research Council in the UK, and the Chief Scientist Office in Scotland. I'm going to finish now. I'm very happy to take any questions any of you have. Thank you.
Thank you, David. This was a great presentation. May I ask you the first question, which is quite clinically? I observe in clinical practice that individuals or patients with kidney failure or also declining kidney function, they are looking older. Can you tell us a little bit about something about the aging process of individuals with kidney disease? Is it just the kidney aging more faster, or are these also are other organs affected?
Oh, well, I mean, I would say chronic kidney disease is undoubtedly a systemic process. And actually, it's a very small subset of patients with chronic disease will die from a direct kidney failure process. But what they have is a huge excess of cardiovascular disease. Actually, it's worse than smoking or worse than being diabetic in terms of your excess cardiovascular disease. Strokes, I mean, it impacts adversely on liver fibrosis as well, frailty. We have a huge excess of peripheral vascular disease. So it's the kidney, which is less than 100% of our body weight, is impacting on many, many different cardiovascular sites here. So yes, I would say it's a hugely systemic process.
And what is then the role of senescence? You very nicely showed that senescence pathway or cellular senescence pathways are upregulated across organs. I think I saw a liver and a kidney, and you saw commonalities. So what role plays senescence compared to all other disease mechanisms?
Well, I agree. The first thing I must acknowledge, I think this is a really sort of multifactorial process, and you can pull graphs from excellent reviews giving, 10 different routes by which this will alter, because you retain fluid, you develop hypertension and cardiac hypertrophy. I mean, people, and many of them, senescence will not feature at all, with that disordered calcium homeostasis as well, with disorders of your parathyroid glands and things as well. And I'm sure all these things matter, and treating blood pressure is undoubtedly helpful. It's very well validated to improve things. But having a patient with absolutely normal blood pressure with maximal dose ACE inhibition, they still have excess risk as well. And my own view is, I think one of the things we're doing nothing about at all is cellular senescence here, and I think there is scope to do that, to try and improve our patients' outcomes. There's certainly a huge unmet need for that.
Yeah. So you showed one graph or a couple of graphs there. You correlated the number of senescent cells with kidney function. That was, I think, in diseased individuals already, because they had a reason to take a kidney biopsy. Do we have any idea how if the number of senescent cells is declining during the aging process in individuals who do not have kidney disease?
Well, we do accumulate senescence in the kidney with aging. I mean, in multiple organs with aging as well. We are limited in our ability to take samples of this, but people have looked, for instance, at live kidney donors. You're getting a biopsy at the time the kidney's going in, when it's had no insult, no ischemia, but as a sort of baseline to help guidance. And yes, I mean, again, it's like a sort of, undoubtedly a highly significant increase with, and again, in post-mortem samples as well. And I mean, it is one of those things that actually in a patient who has been well all their life, but then dies suddenly and donates a kidney, we recognize someone in their 7th, 8th decade, that kidney doesn't do as well. It doesn't survive as well. We try and match it to all the recipients, because there's just, an unidentified sort of something in the aging milieu that is different. We sometimes get reports on kidney biopsy like that its kidney biopsy is normal for age, which is not the same as saying it looks like an 18-year-old's kidney, because it doesn't. It's different. And more fibrosis is, they say more fibrosis is physiological in old kidney, but I suppose, again, the question is, why is it there? We don't know. And I would, as a sort of senescence fan, would say, well, perhaps one of the things is that you've got more senescent cells and they are quietly driving it in the background.
Okay, then I will repeat my question. What do you think is the contribution of senescence in the aging process of decline in function in kidneys compared to all other factors? Any idea?
Oh, goodness. Well, I mean, I believe it is significant. Significant, perhaps even major, on the basis that actually is when we've treated that, you can optimally control cholesterol, optimally control blood pressure, and you still have major morbidity, which remains unaccounted for. How much of that is senescence? I mean, it's only going to be by taking, effective, safe drugs into our patient and testing we actually get to see.
Yeah. Okay.
Which is something hopefully down the line we get to.
Yeah. What you really nicely showed is the kidney after transplant, and I think it was a rejected kidney, that huge accumulation of P16 positive cells. So what is the reason, and how often do you see that in clinical practice, that if you're transplanting a kidney, that you have a rejection of that kidney? And how often do you see such an accumulation of P16 cellular senescent positive cells?
I mean, rejection clearly is a big problem for kidneys. The answer is actually, how often do we see it? We never look for it. Those were research samples they ran, and actually these kidney biopsies we're running at the moment, I mean, have all been run. They were taken for clinical reasons, and nobody looks for P21 or P16 in them, and we're detecting that now and trying to go back to relate it to outcomes with that as well. Actually, another thing, I mean, because the Melk and Schmidt groups have done some great work on this, and one of the things they did, which I thought was really impressive and persuasive to me, was that when they look at kidneys from mice lacking P16 and P21, they survive better. So again, I just said that was very helpful information to say, actually, this is not, because I think that graph is, or image looks different, but it could all have been due to something else. But the fact that actually these kidneys seem to function better and survive better when these pathways are taken away, again, would I think be very consistent with the work we've produced, and another group actually from the Mayo Clinic have published very recently as well, again showing when you deplete senescent cells using a completely pharmacologically distinct treatment, they also see protection.
Yeah. Final question before going to Jessica Liu. Really getting all the questions from the audience into this discussion. As you know, senescent cells are not senescent cells. So it's a huge heterogeneous pool of cells being either repetitive senescent or stress induced senescent. If you would have to choose which kind of cell to remove out of your patients, maybe in 10 years time when we have treatment, where would you put your money on? What kind of senescent cells and what maybe cell type even?
Yeah, well, I mean, this is something we're learning a lot about at the moment through single cell data. And as I hope I can convince sort of a person or two at home from one of my last graphs there, and I mean, it's very recent data actually we've had in the last couple of months, is that not all senescent cells are the same. They're producing very different parameters of things. And yeah, so I have strong suspicions that you've got some are, actually behaving quite like that archetypal acute senescent cell, not producing anything very bad, signaling for their clearance, but there are others that are really are real bad guys producing different things which are going to keep stuff there. Those would be the ones I would go after. And I mean, I think.
So which are the bad guys?
Sorry?
Which are the bad guys?
I think there's, I don't want to mislead anyone, and we don't have all the answers for that yet. What we're in the process of doing is we've got long-term studies looking at which ones we can hit. And I do hope that actually can turn into novel, really clean treatments you can deliver to patients to try and knock out these bad cells.
Yes. Okay. Over to Jessica. Jessica is a PhD student in our group. Really fascinated by digital phenotyping. But Jessica, what are the questions from the audience?
Thank you, Prof Andrea. Thank you, Dr. David, for your presentation. So I think we'll stay on maybe kidney transplant topic for a little bit. One of the questions is that in clinical practice, what is the maximum age that a donor can be to donate a kidney that can be transplanted into a recipient?
Yes. Well, as far as I'm aware, I think there's no absolute set, certainly in the UK, no absolute set in stone figure on that. Having said that, and I mean, we had an altruistic live donor aged in her early 80s, I remember, in our unit a few years ago. I have to say there was a bit of sort of a few sharp intakes of breath when this was announced that's what was happening. And I mean, she was in exceptionally good health. There's sort of a swing, and I think that occurs, after the age of 60 to 70, where you realize these kidneys are more vulnerable. They're less likely to work straight away. They're more likely to develop fibrosis, which is one of the bad things with it. And I mean, the trouble is, or perhaps is an opportunity as well, is that dialysis is so bad for you that even having, if you like, a sort of suboptimal kidney is better than being on dialysis. And there's a limited window where patients sometimes are fit enough to receive a transplant. And one of the things that we have done and where clinical practice has changed is that we recognize that some of these less good kidneys are still maybe the best option for a patient compared to not getting one at all. So we're transplanting more kidneys than ever before. That's mainly because more of them are expanded, what we call expanded criteria donors, ones that might previously have gone just for research or just gone in the bin are now being transplanted.
And to follow up, is it possible that a kidney from an older individual, like that lady who was 80, may transfer senescent cells into another individual during a kidney transplant? And if so, if it's possible, is it a problem? And if it is, what is currently being done about this?
Well, I mean, I think it's a very good question. I can't give you a definitive answer to that. I don't think anyone can just yet. I can give you a hypothesis or a guess would be, I think that receiving an organ with more senescent cells has a potential. My belief is that, and we have some evidence which is not yet published, that senescence will transmit through the circulation in a systemic manner. Now, it's not to say that it would be whoever receives that kidney is making a terrible mistake, because actually being on dialysis is very, very bad for you. And again, it's another thing we are starting to look at, and I think it would be brilliant if it would work, could you, for instance, take a kidney, give it a dose of a medicine to kill its senescent cells selectively, and then transplant it? we're moving to systems now where we have normothermic perfusion, where a kidney can be kept alive and perfused for a few hours waiting for transplant. And that perhaps gives you a window even to use a drug that perhaps, say, a drug that would reduce your platelets if you're taking it orally for a month. Perhaps you give it for a few hours. And based certainly our mouse work, if it translated, would suggest that actually that kidney might well behave better, might regenerate better for that.
Wow. And then the next question is, when and what part of the kidney do we start to observe senescence during aging? Because I know like there are different parts. And is there a kind of logical way that it starts kind of turning to senescent cells in the kidney?
Well, I mean, certainly based on our sort of our study of biopsies and our study of mouse kidneys and things as well, it seems to be the more distal tubular segments are the bit are most vulnerable to it. And it may tie in with the fact that the kidney, the way its blood supply is divided, tons of blood goes through a kidney, but most of it is whizzing in and out again through the glomeruli. And some of these cells down there are actually quite hypoxic under baseline conditions. And I think under further insults, we'll be receiving the most hypoxic insults and things. But certainly that's where we see the biggest burden of senescence.
Have you, your group, or any other groups seen a gender difference with regards to the level of senescent cells that is observed in the kidney during aging?
Well, in some of our own work, what we find is that females tend to develop less senescence for the same insult and actually have better functional outcomes as well. I would have to chunk back through the literature to see whether that's mirrored in that as well. Certainly, actually, when it comes to outcomes of kidney injury, females, certainly at least until the menopause, are very much protected. They're much tougher. They develop less injury and recover better.
Do you think that sort of affects, like, being a specific biological sex also affects kind of kidney aging the same way it might affect other organ aging? And how would kind of senescent cells or aging in the kidney kind of affect aging in other organs as well as it kind of?
Yeah, well, I mean, again, I suppose my clinical experience and my sort of research interest make me think that having more senescence in any organ is a bad thing and has the potential to transmit in the way that, your arterial tree will wear out more quickly. You're more likely to get a stroke, more likely to get heart failure if you have a kidney problem. I believe that is transmitting. I mean, so another thing actually which intrigues me is patients with failed kidney transplants. I mean, the practice generally worldwide is if somebody has had a kidney transplant and it has rejected, then you withdraw immunosuppression and try and sit in it, as it were, to avoid an operation. About 50% of the time that's successful, and other ones they have to have them taken out. An intriguing thing actually was published in JASN a few years ago was that patients that retain a sort of, if you like, a fibrosed senescent kidney transplant in them, they have higher mortality on dialysis as well. Now that could be through subclinical rejection, could be because they were less healthy anyway, but certainly, again, as a sort of senescence fan, I think I'm intrigued this sort of is it worse having a sort of fibrotic senescent focus there than nothing at all? And again, actually something we've done in our work is comparing injuring one kidney versus taking a kidney out. Because one of the arguments is, well, actually, when you see chronic kidney disease, it's just because waste products. You've got more waste products in your system, and that's causing it. And that's quite possible. One of the things we tried to do to address that was, well, let's take a kidney out. You lose 50% of your function, versus leaving a kidney in that's still helping a bit, but you've got senescent cells there and fibrosis and stuff, which will be better for you. And actually, you see changes throughout the body. You see changes actually in the healthy kidney when you've got an injured one there that you don't if you take one out.
One of the
Again, we're still. Oh, sorry, go on.
No, sorry. I guess one of the follow-up questions and top voted ones are, what are the differences between injury-induced senescent cells and aging-related senescent cells in kidney, which maybe follows up from your previous?
Yeah, well, I mean, I think that is a very interesting question, and it's one that we don't have a good answer to yet, but we are in the process of trying to do that. Because I would certainly, I suspect people are wondering, are all senescent cells the same? And I certainly don't think they are in that. And the question is, how do all these differ? So I mean, something we are in the process of looking at the moment is how does a really fresh senescent cell differ from a really sort of a long-lived senescent cell? And actually, how closely, because we have this genetic mechanism we can switch senescence on, but is that actually the same as what you get in the aftermath of injury or the aftermath of aging?
Right.
I mean, the suggestion is there are differences there. What we need to do is really try and tease out and look at that in a really robust way to come up with an answer.
Right. Is it possible that there's a safe baseline number of senescent cells in kidney that won't affect overall health? So like you injure it a bit, but you know, you don't have enough senescent cells to really kind of affect your health.
Well, it could, I mean, it certainly could be. And I suppose in particular, if there really is a very benign inert senescent cell in there within the renal epithelia, it could be, I have to say, maybe again, as a real sort of senescence groupie or something like that, my inclination would be that, you know, when you look at healthy young mice, healthy young patients, they've got virtually none. And it's certainly not bad for you to have no epithelial senescence at all. My inclination would be that the lower your count, aiming for 0, would be safe and fine.
And I guess a related question is, since we're talking about mice, what are the limitations of mice as model organisms for the study of senescent cells in kidneys?
Well, I mean, clearly, a mouse model is just that, it is a model. These, you know, the injury we deliver here is certainly not the same as a sort of a multi-morbid sort of setting we're classically seeing in our patients. We have an old patient with diabetes, with other conditions coming in, with a GI bleed. There's lots of things happening there, lots of medications which we're not replicating. So we have to bear that in mind. I mean, one thing we've tried to do to try and tackle that, because actually 99% of all the research done in mice is done in mice about 8 weeks old, which are, you know, probably like late teenagers in terms of study. These would be an example of the most IRI resistant, sort of most adaptive repair prone sort of animals anywhere. And one of the things we've done, which costs a lot of money and takes a lot of time, is age mice out to 18 months. So look at that. And an 18-month-old mouse behaves differently. Indeed, even a young mouse that's been irradiated, such that it's got a senescent cell burden, it behaves differently as well. In the same way, actually, a patient who's had leukemia, has survived it with total body irradiation, they have about a 1 in 3 risk of developing chronic kidney disease. You know, with no underlying, you know, they talk about radiation nephropathy as a condition where you look at it, where you've got scarring and lots of radiation-induced senescent cells.
Wow. And knowing kind of what we know from tonight, are there any diet or lifestyle changes that we, as the audience, can kind of take or adopt into, you know, our everyday life to help keep our kidneys young or more resilient to kind of aging?
I think my sister is a dietitian and would be much better at answering that than I am. I mean, I think what evidence there is out there is maintaining a healthy weight is good. Obesity sort of drives senescence there with that. And I think there's also some evidence from the La Brazil group over in the Mayo Clinic that actually physical activity is good as well. That actually, if you have even mice and exercise them regularly and things, this correlates with lower senescent burdens, and that correlates with better outcomes as well. I bet there are good medicines out there and good foods that we should be taking to do that, but I'm afraid I'm just not expert enough to give you a good answer to that, I'm afraid.
No, it's good. I think it kind of reinforces that a lot of good lifestyle habits and eating habits are not good for, you know, just kidneys. You
can see my bicycle parked behind me at my fireplace there, actually. It's my effort to try and avoid getting too obese here.
I guess to wrap up, I guess what are some of the kind of future research work or goals you hope to see in terms of kidney aging and senescent cells?
Yes. Well, I mean, I suppose going for a blue sky approach, what I would love to see, I think it might be in 5 years or 10 years, would be a setting where we were giving medicines to suppress senescence, to kill senescent cells safely or render them absolutely inert, to then hopefully make not in the kidney, but for many organ systems to allow people to live longer, healthier lives. There's a few hoops to jump through before that. I think we need better biomarkers, so we actually have a way of being able to assess how much senescence do you have and how much senescence will you have after this treatment. We need to get these drugs tested through their murine models and into human practice. But I think that's got to be the goal for us all as researchers and clinicians.
Thank you so much, David. Thank you so much, Jessica. This was a wonderful discussion. At least what I learned is we might be able to rescue the kidney function by attacking senescence.
One day, I do hope so.
Maybe by more physical activity or even biking. But David, you said senolytics before transplants. So if you have the first results, let us know. We will find a slot for the show. So we should be the first ones to know. Thank you so much for this wonderful presentation. I hope you, as listeners, also enjoyed today's show. Use the panelists and all attendees' option to leave comments and feedback and questions, and we will have a look at that. Follow us on our new Twitter account. Today we have 500 followers. We were very, very glad and celebrated that a little bit. But please be number 510 and 2000, etc. Also, check on our LinkedIn account and on our website. You will find the links in the chat. Next week, and that's the 20th of October 2022, Professor Cleo Bishop from the Queen Mary University of London will be my guest. And she will not talk about TikTok, but she will talk about TalkTick of senescence. So we will stay with the topic senescence. As always, there is a final video, and I would say, and summarize, just keep working and enjoy. Take care.
My name's Beryl Carr, and I'm 100 years old. I've been working in the League of Friends Cafe for 18 years as cashier on the till. I started volunteering here when I moved down to London when my husband died, and I didn't know anybody or anything, and this was my lifeline. My daughter made inquiries, and I've been here ever since. The friendship and the camaraderie, and I feel I'm helping people that are not as fortunate as myself.
It's absolutely amazing. I've been at Ealing Hospital coming up to 9 years now, and Beryl has been there. And Beryl's always here. When we come at lunch for a cup of coffee, she's there with her smile, and pre-COVID, often a hug as well. And I think she's a complete inspiration. Many of us look at her. She's always got a smile on her face, a swagger as she walks.
I can't believe I'm 100, but I am. Celebrated with a big party, and I've had several smaller parties since then. It's been going non-stop for the last week.
It's like having a bit of royalty because everybody comes and sees Beryl when she's here. They make a special point of saying hello and making a fuss of her.
Oh, I couldn't believe when the postman came with this card. I was overwhelmed. When I took the picture out of the envelope, I saw how lovely she looked. I thought how lucky I am to get it. I can't believe that I've reached this age, but obviously I don't feel 100. There we go. I'm not ready to put my feet up yet. I've got a lot more volunteering to do. I'll be carrying on as long as I can.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.